Hi everyone, let's get started. So we are towards the tail end of the course. I saw some of your project proposals, submissions, and it's looking good. Keep making up the progress. Today we are going to be talking about the human angle in lossy compression, particularly focused on the vision aspect of it. So it's another sort of special topic, and it's going to be a fun lecture. It's kind of stuff which is typically not covered in information theory, but it still finds its ways into like practical lossy compressors and stuff. Yeah, any questions, by the way, so far on learned or logistics or anything? Cool? No? Okay. So yeah, so far in lossy compression, we have looked at a lot of different things, right? So we have looked at quantization, which is a core mechanism of introducing loss. We have looked at third underpinning of a rate distortion trade-off. Homework 4 also has a question to like kind of get you warmed up and practiced about it. We have spent some time looking at optimal solutions in the case of Gaussian sources, which builds up our intuitions, as well as particularly focusing on MSE distortion, right? Like so far in the course, mostly we have stuck with MSE. And then we talked about image compression and JPEG and learned image compression. Okay, so this class is going to be very different. So the main point is that like all multimedia, at least, eventually is consumed by humans, right? So it's for the human consumption, and human senses play a big role in the design of these lossy compressors for multimedia, and we are gonna understand like what properties of this sort of human sensory perception comes out and plays in our design of compressors. So in particular, we'll look at questions like, so why some of the design decisions were made in the image compressors we saw, like JPEG in particular. Or how can we further improve image video compression, knowing the fact that, oh, this content or this whatever signal is eventually supposed to be consumed by us, right? And not just by machines. Finally, I think we have touched in a little bit about it, but want to just give you guys a flavor on like, okay, MSE is not ever encompassing, right? So what are some other options which you might see if you would practice compression outside? Okay, so this talk is derived from lots of great resources. Feel free to check them out. So even before we go into images, so this is really like, so teaser number 1, okay? So what I'm showing here on the right is I'm using this tool, Media Info, and this is a compressed Pokemon theme.mp3 file. What this Media Info does is like it tells you various stuff about this compressed file. So for example, file size, duration, so on and so forth. Here you see the compression format is like MPEG audio, which is like parallel to JPEG. It's just omnipresent. And then if I keep going down, at some point I see 2 channels, and I see sampling rate of 44.1 kilohertz, right? Why do you think like this file or typical audio compressors have like 2 channels or this particular sampling rate? Yeah.
Probably because human hearing goes to like 20 kilohertz or so, so 44 would let you rely or accurately produce up to like 22 kilohertz.
Yeah, so okay, as answered, like he said, human hearing goes up to 20 kilohertz. That seems to give a hint. That's exactly correct. So the 2 channels is because we have 2 ears. If humans had 3 ears, maybe this file would have been compressed with 3 channels, and that would have made better sense. So this very practical design decision, right? So this like, if I keep this file uncompressed, it would have, you are keeping 2x the number of channels. This very practical design decision was taken into account, like very simple fact, like we have 2 channels, and sometimes you want like stereo sound in your stuff, right? So this is why 2 channels. And 44.1 kilohertz is exactly again what Sean said. So if you, I don't know, go back to your elementary biology, like you might have heard that, oh, humans can hear from 20 hertz to 20 kilohertz. Though in practice, I don't know any human who could actually hear 20 kilohertz. So if you can, that's great. And so if you know about Nyquist criteria, you would need 2 times the highest frequency to represent the signal without any aliasing. And that's where the 40 kilohertz come from. And then 44.1, it's even more fun. So you want to give some buffer because you don't have perfect low-pass filters, which is typically required in these sampling techniques. So you had to give some buffer over 40. And then 44.1 in particular is very interesting because it can be factorized by 2, I think even 3 and 5 and multiple different primes. So that allows you to later on downsample this signal by throwing away samples much more easily. So even this innocuous, very random looking number, it didn't magically come out. People thought about it, sat down, and then like, this is now default. So almost every file, like you don't think about it when you are saving an MP3 file, but these design decisions are there and they are determining the file sizes, right? So I could have had double the sampling rate, half the sampling rate, and that would have completely changed my compression and file sizes and the kind of reconstructions we would have had. So very direct example of how humans are playing a big role. Like just the idea that humans are going to consume this plays a big role. Okay, then teaser number 2. I have already asked this question. Maybe let me ask it again, or I'll just go through this fast. But this is really a very iconic picture because it very clearly suggests what's happening in the image compression case. So here I have some source image. And all this B, C, D, E, F images basically have the same mean square error with respect to A. But clearly, if you look visually, for me at least, B is much, much better reconstruction than any of the C, D, E, F. For example, C is like smoothened, E is extremely smoothened. D and F have just clear noise sprinkled over, right? So again, this clearly says that not only that a lot of design decisions were made with humans in mind. If you are not careful about designing your compressors in future without taking that into account, you could have designed a compressor which would have resulted in reconstructions like this, which is just bad. That's a bad design. Like nobody practically would like to keep a compressor which generates reconstructions like that. So it's also important to understand really for the design, practical design of compressors. Okay, so rest of the lecture will just get some preliminary understanding of how human vision works. We're going to focus on the visual aspect of things. We are not going to talk about the audio. We'll see its role in the design of traditional compressors, learn a bit more about perceptual metrics, and the last part would be like how do you account for perceptual metrics eventually in this rate distortion kind of framework. So let's get started. Again, it's going to be fun, it's light, so it's going to be different, so do interact. Let's see how this goes. So this is just like a typical human eye anatomy. So here you have some cornea pupil, your eye lens, which basically focus any optical signal into the back of your eye, which is this retina tissue. Here at retina, basically, you have transduction, which happens, which converts these light signals into electricity. And finally, like through this optic disc, through your optic nerve, you send back these electrical signals back to the brain, which then process and makes you see different things. So right here is our first thing. Like when you actually have a light source which is coming, suppose you have a light source which has a single intensity. So suppose you had a delta function in terms of pixels, right? Everything is black, 0, and then just one pixel having very high intensity. When you look at back of the retina, it's no more a delta function. What happens is that this line function spreads out. Okay, so you have like a low pass filtering which your eye is doing right at the beginning, just because of the physics and optics of things, even before anything else happens. So this is important because this kind of goes back and correlates to our fact, right, that you can throw back high frequency stuff. That's because, hey, if there were like many of these lines close to each other, I wouldn't have been able to distinguish them anyways. So there is no point of keeping extremely high frequency stuff. And interestingly, like you can argue like nature also has signals which have very low power in the high frequency spectrum, right? And you can always argue like humans were designed to pick up signals like that versus like this is just like the basic optics of things, right? So it's interesting. Right there you have something which you can utilize in your compressors as soon as you understand this very basic thing. So this is a bit more detailed. Don't have to worry about it. This is just like the blown up part of this retina. what you need to basically think about is this. So suppose you are watching your favorite Tom Cruise movie. What happens is the light kind of falls in this first region, which is your photoreceptors. So this is just like blown up part of retina. These photoreceptors are what transduce this visual signal into electrical signals. Okay, so you have light to electrical conversion, like the wavelengths to electrical conversion, physical property to this electricity. And then you have a lot of these neurons, which are called RGCs or retinal ganglion cells. Again, don't have to worry about the names and details as long as you follow through the ideas, which are basically these spiking neurons. So these issue like electrical spikes, which are then sent back to the brain, which processes these electrical spikes and bring out some visual picture for you, like whatever you are seeing in this real world. And so here is the second big difference, sort of compared to how we think of images versus how humans process it. So we kind of treat images as an array of pixels, right? Like that's what you have seen, that's what you probably just think. If I ask you generally, like sitting around, like what is an image, you'll probably say, oh, 3 RGB values, and like that's basically an image, right? But humans are not really perceiving it like that. What happens in humans is that you basically have many different kinds of these retinal ganglion cells, around 20, and each of them are basically independently tiling the whole screen, whole scene. So it's like, think of this as like one set of retinal ganglion cells, yellow ones as the other, green one as the third, blue as the fourth, and so on and so forth. And you can think that each of them basically sends an independent signal to the visual cortex. So it's like you have, it's debatable a lot of things, like we don't know very precisely, but this is still a relatively advanced field of neural. Different cells are basically extracting different features of the visual scene and then sending it back to the brain, right? So which is very different than how you would think of RGC, where each pixel is basically doing one thing, and then you build machine learning models or whatever it is to extract different features. In humans' case, like it's like you already have these feature extractors in some sense, right at the beginning, right at the retina, right? And then you, on top of it, build using your visual cortex, like much more complicated associations in your brain. So again, like the difference is human retina is not your camera. It's not just pixel array. It's doing something very different, the back end. In particular, I think these rod and cone cells, again, like a lot of this is even at GK level you might have heard at some point. But it's very interesting because these do decide a lot of interesting things. So these rod and cone cells are really important because these are our photoreceptors. These are our sensors. This is how we are sensing the physical signal coming into our eye and making some interpretation out of it. And these are actually very, very amazing. Okay, so first of all, rods, they are responsible for encoding intensity. So their only role is to figure out like what's the intensity of the scene which is coming in. No color information, nothing else. And these are amazing because they are like quite a number, like almost 100 million. But even more than that, they are able to adapt so as to see almost a dynamic range of 10⁹, like almost a billion orders of magnitude. What I mean by that is like, think, if you are a human, you can be sitting in outside California summer weather. After a while, you can see everything there. And then suddenly you have a class, you rush into your class and it's a dark room. Okay, initially you are not able to see anything for maybe a few seconds, but within a few seconds your eyes adjust and you are able to see everything in this room. Now think about like what's really the physical signal which is coming into your eye, right? When you are outside in the sun, your intensity was extremely high, right? And then everything else which was happening was around this base level. Whereas when you came into this dark room, suddenly the intensity fell significantly, right? And then you were basically seeing stuff around this level, okay? And this is a hard problem if you think about it, right? Like our cameras have struggled to adapt to it till date. Like, I don't know, iPhones, one of the selling point is great night pictures. Right? Like why? Because it's hard to detect things at that level of intensity level. And we as humans, we just do it. And we don't even realize, right? When we are doing that. And rods basically have, they are the contributors. Their only role is to adapt to intensity, figure out, and change sort of like the base level around which they are gonna kind of see the fluctuations and see things around you. Okay? And another very interesting thing about rods is, so if you look at this diagram here, you can think of your eye as like a spherical surface, right? And then there are like, you can basically put angles to the retina of where things are happening. And very interestingly, you have some optic nerve, which is sending signals back to the brain. But where this optic nerve connect, you don't have any sensors. You don't have any retina. So whatever falls in sort of this region, you can't detect it. And it's like a fun exercise if you want to do that, like, I don't know, I find it fun. You kind of just focus on your thumb and you keep seeing straight and keep moving your thumb at around, so I can see my thumb, I can see my thumb, I can see my thumb, I can see my thumb. It just vanishes for me. And then it comes back. So you can try it now, try it at home. That's like a consequence of having this blind spot, basically. That at certain angle, I just have this blind spot where I can't see because all the light there is being focused in this region. But even more interesting than that, so here is like y-axis shows the density of these receptors. So you can think of how many sensors you have in that region. If you look at the solid line, the rods, so they are present almost everywhere in the retina except like this region, which is your blind spot, like around whatever, 20 degrees. The other cell type, which are called cones, on the other hand, if you see their density, cones are basically not present almost in whole of retina except near this almost 0 degree angle. Okay? And the role of cones is exactly opposite to rods. So rods are just basically trying to understand the intensity, it's giving you the dynamic range. Cones are responsible for colors and details and all those things, right? And they are completely absent in the retina except for maybe a very small part of your central vision. Right? And this central part is called basically fovea. Again, why is this interesting? So if you think about it, if we only have high acuity or high resolution vision in such a small part, like if you think about it, this is just like a few angles, you can convert it into physical angles. It's like really small region of the physical world which you are sampling with sort of this high fidelity. Like other places you are just encoding like a grayscale kind of level to get adjusted. So then how am I able to see this sort of scene in a very smooth and nice fashion, right? That's an interesting question to maybe think about. And the reason for that is we are actually never seeing any static scene. So when you think you are like just looking and you are seeing an image, that's not really true. You are actually not seeing an image. At any given point, you are seeing a video. And your eye is not really focusing at one point. What it's doing is, your eyeball really, it's not eyeball, sorry, the eye in general, like it's doing these movements called saccadic movements, which is just basically like it's moving really fast. Like you don't even know, but it's continuously doing this. The reason it's moving this fast and it's doing this is to basically keep changing the point of the physical scene, which points to my central region, my fovea. So think of it. It's like there's a very small point at the center where you have high resolution. Everything around it is low resolution. Okay? So when I'm looking at this scene, if I just do this, I don't know, right now I'm just completely focusing on clock. And actually all of your faces, all of this thing has really vanished for me. I can't see it if I like very consciously focus, right? But if I'm being subconscious, I'm not consciously putting my eye to say, okay, just look at one point. What my eye really is doing, it's sampling this region, but it's not staying there. It's like microsaccades are in like milliseconds case. It's basically just continuously moving and sampling. And then your brain is interpolating all this signal to form this continuous kind of smooth looking picture. Right? So again, like a lot of fun facts. This class is going to be a lot of things like that. So it's pretty cool. Like when I first learned about it, it was amazing. Like my eyes are not staying at the same spot. But now you can be like, Pulkit, why are you talking about this, right? Like there's so many GK questions, you can do so many things. This idea that you have very high density sensors in a single place. It's actually called foveation. And it can be exploited in some very interesting ways in your design of physical sensors and how you do compression. Okay. So for example, what it says is that at any given point of time, I'm only sampling small region in high density. So then it makes me think maybe we can exploit this in some way, right? Maybe if I am able to track human vision, let's say, then I can, if I know, right, okay, at this particular second I'm just looking here, let's say next second I'm looking here, and if I can work at this latency and just track my human eye is going to look more densely at a particular part of a scene when you are watching your favorite movie or anything it is, then I don't need to give too many bits to everything else, right? I can just give more rate to wherever my humans are focusing, and I can give 0 rate to things outside. And this may sound very sci-fi, but there have been over past few years, there have been lots of interest and research projects trying to exploit exactly this idea in the VR setting. So right now we are only focused in 2D setting, but now you can think if you have a VR headset, maybe we can do something to actually track where humans are looking. And maybe we can figure out how to distribute bit rates, because in VR you now have a 3D environment, right? You don't want to give equal bits to everything on the periphery. So the kind of things which we are learning also have implications for future technology, right? It's not just things which we have right now. Lots of cool projects along this basic idea. Yeah, I think we talked about the first one. Just like if you have seen this before, so the adaptation aspect of human eyes, it's basically called Weber's law, which basically says that we only care about relative changes, not the absolute value of changes. Again, very small point, but it's very interesting how you can exploit it in your signal processing and compression aspects. What it says basically is a bit change at a high value of luminance value would not really, it won't be as significant as a single bit change at a low luminance value, right? Even though it's exactly 1 bit change. So you need to be careful about when assigning rate and all those things in these situations. Or maybe you are uniformly assigning the rate. In that case, you may have different artifacts which might be visible to you just because those artifacts are occurring in low intensity region versus the high intensity region. Yeah. Again, this aspect, maybe it wasn't so important 20 years ago. But we are in a world where we have 4K TVs, HD, 4K, I'm pretty sure we are not going to stop there if we can fit all that data and be able to process this. We want million more pixels in our cameras, million more pixels in our TV, right? And at even 4K HD level, all of these effects starts becoming very significant. And the big companies spending big money trying to understand these issues and improve upon them. Because it's straightforward money, right? Each bit saved are your user experience. So simple idea, but can be exploited in interesting ways in engineering setting. Okay, I'll pause for a second. Any questions? So the next thing is, so we were just talking about cones, which look at colors. What this plot shows is on the x-axis, it's the wavelength, which is how light is colors are represented physically, right? Colors are just different wavelengths. So 400 to 700 nanometer is roughly our visible range, 350 to 700, I think. And then y-axis is basically the response for these cones. At different levels of wavelength. So think of these as sensors. These different sensors respond differently to different wavelengths. Okay? And then what you see is that you have dots which basically respond to roughly it's not really it's something, but it's roughly responding to the average wavelength or in the visible spectrum. But then you actually have 3 cones. And these 3 cones are sometimes called LMS, just long, medium, small. L for long wavelength, medium is for medium wavelength, small is for small wavelength. So that's the neuro terminology for these. Now, why do you think you have 3 cones? Do you think that makes sense? Or is it something confusing? The answer is on this slide, by the way. don't even have to think that much. So I'm sure at some point you would have wondered, oh, why RGB? Where did this RGB thing came from? Like, at least I used to wonder quite a lot. Like, why RGB? Why are we stuck with these three colors? And that actually comes down because your physical, like your human retinal sensors, cones, actually come in three flavors. And they are roughly sampling the red color, the green color, and the blue color. And that's why like RGB is one of the ways you would like to represent, because back in 1700s, a lot of philosophers, where you couldn't actually do these experiments, through other psychovisual experiments, figured out that, okay, maybe RGB seems to have some special place in our vision. And that basically later on in neuroscience you could even figure out like their exact response characteristics, so on and so forth. So again, like even something like RGB it's actually other way around. Like RGB doesn't come from here. Like since we have these things, that's why RGB has found like a special kind of place in our thinking about visual images. And so this RGB color model, this is also sometimes called trichromatic theory of color vision. So fancy name, but just English, trichromatic theory. But interestingly, trichromatic theory is not the only theory of colors, right? Some point you might have, so these are two physical colors which are exactly same. I could have represented this as RGB, like some different values of RGB, which gives me a certain color. But maybe you have heard of a color space called CMYK. Probably not. So this used to be used in old school printers with toners where, so RGB is like an additive color space where, oh, I add R, I add G, I get a color. In this case, like these values of RGB gives this color. But I can also think of it as like CMYK, which are sort of like subtractive colors. So when you add CM and Y, you get like a black. So it's like removing sort of thing, but it's the same color. So nobody said like, oh, I need to represent this as just this RGB. I could have represented this as CMYK. And actually on the right, what I'm showing is, this is actually a very complicated and not so obvious and confusing topic for engineers, like where does these things come from? But actually there have been a lot of studies on like how colors are represented, not just at the neuro or engineering level, but actually at the psychovisual level. So at the end of the day, this is like a psychophysics experiment, right? Like what colors do you see? Like how can I represent the same color? And there have been tons and tons and tons, like philosophers all the way back, I think, and starting from 1700s, people have been asking these questions, trying to understand them in deeper and deeper details. And this is something which you might hear if you work in image compression, video compression at some point. So the CIE, it's like just a society, in 1931 came up with something called chromaticity diagram. So again, chroma for color. And it looks like it basically looks like 2D space to represent colors. And what you have is like different standards and companies, etc., have adopted very different color spaces. So you have RGB, which we know of. There is something called sRGB. Okay, so first of all, there is something called visible spectrum. Like, so visibly we can only see these colors. So this has a weird space. I'm not going to go into details of why, but if you're interested, come talk to me later. But yeah, so the yellow one is the set of colors which your human eye can see. Okay, and then in practice, you actually can't represent those many colors in the same way as you would like to. So historically, people came up with RGB, then there is sRGB, which is this black curve here. There was Adobe had an RGB, which is like Adobe RGB, which is this one. So Adobe basically came up with its own RGB standard. Now you should ask like, okay, why am I talking about these standards, right? The reason is this 224, 102, 102, right? We simplified our discussion of images and we just said, oh, it's like 3 bytes per pixel, which responds to one color. But that's not true. At the end of the day, these digital values are converted into physical signals using your monitor. Maybe they are printed using your printer. Maybe the actual your camera, when it was recording, it was converting this physical signal to this RGB values. And all of this sort of depends on what's the color space, right? Like, so for example, if a display has a different color space than whatever it was recorded with, you will get very different reconstruction. And that's important for us because that changes the distortion completely, right? So you might have to think about these things. And again, it might seem like, hey, we never think about it, do it, but it's actually very common, and you have some very interesting bugs which come up if you were doing video compression, image compression, if you are not being careful about these very small things. Because like just different data recorded in different way, displayed in different way, and you'll be like, you'll be thinking, why is my Huffman encoder not working or whatever, LZ variant not working, but it will be this color space issue which you might be sitting with. So it's just important to know that these things exist and do play a role. Okay, so this is gonna be fun. So let's talk a little bit more. Let's do this. By the way, yeah, like actually just a quick show of hands. Like this dress I think blew up sometime last year or year before. How many of you see this as gold and white? Oh, okay. How many, what do you see it as? Let me ask, I forgot what's. Blue and black, excellent. So, okay. How many of you see it as blue and black? Like, why is it happening? It's the same RGB values I'm showing here, right? So right there, it's very interesting, and we'll motivate this for the next thing. But even before that, let's do this quick experiment. I think. Oh, yeah, it loaded. Okay. So what I want you guys to do is, okay. First of all, what do you see here? Don't focus anything. Just look around. What do you see? Anyone? Well, see something. Okay. Do you see a vanishing purple circle, sort of, right? Okay. Now focus on the X, just on the X. I just want you to focus on the X. I don't know if this is going to work in this room or not. Let me also come here. Oh, what happened? You see a yellow dot? Okay. Anyone else? You saw a whole circle of yellow dots? It's interesting. Okay, I would have called that color green, but maybe it's yellow. Yeah, it's like greenish yellow, right? So if you keep focusing on that X, the vanishing one suddenly becomes like a greenish yellow color. Like again, it's not your vision. It's nothing wrong with your eyes. You don't have to go get new glasses after this. It's a known effect. And that happens because, so we talked about the trichromatic theory of color, but there is another theory of color, which is basically called this opponent process theory of color vision. Okay? Which basically said that there are pairs of colors which are negatives of each other. So the effect, by the way, which I showed you in the last slide, it's called the after images effect, which is like if you focus on one color, it's like your eyes are adapting to subtract that color out. So suddenly when you look at somewhere else, your mean color is somewhat very different. And so that white space in that previous lilac chaser example suddenly becomes like negative of red, which is what you sort of saw. Like not really negative of red, but which is that yellow, green, whatever color you guys want to call it. Right? So that's like again, a very basic aspect of how our human vision works. And that's the sort of origin of this YUV thing, which we have talked about in the class before, right? Like we had this random color transform, which we said, okay, let's do YUV. We are not going to work with RGB. And I said, okay, later on we'll talk about a little bit more about this. And it's basically this theory of color vision, which suggests the idea of this opponent colors, which basically says that, oh, maybe we have this blue, green, and red cones. The way humans perceive it is actually not these signals, but you merge them. You merge them in a sense where you get this white and black signal, which is basically somewhat summation and addition of all these three wavelengths. White is presence of all these three, black is subtraction. Then you have a blue, yellow, and then you have a red, green opponent colors. And it's debatable. There have been lots and lots of empirical evidence on all sorts of axes. We haven't really finalized on what is correct. But it's like even very recently, I think 2019 or 20, there have been papers that say, hey, this theory is wrong or this is right, like here is more evidence. So, and this has been going on since I think 1800s. So it's very interesting. But for better or worse, we are stuck with this, and this shows up in our engineering applications all the time. So again, like just recall our image JPEG compression, right? So we started with RGB, and the first step was transformation. And then we also briefly touched upon that we are gonna downsample the CBCR components or UV components. And then every DCT as standard thing. Now we can understand this better. So why CBCR or UV, like all of them are, consider them equivalent, like they have some historical differences, but for today's class, assume why UV, why CBCR. Sometimes you'll see like star marks on top of it, like literally like this, CB star CR, oh yeah, here, sorry, Y dash UV. Sometimes you'll see like Y dash UV, Y dash CBCR. Consider all of them equal for now. There are slight nuances and differences which we can talk. But the main reason for, maybe if we think about CBCR, it's very obvious. Why going for the Luma component, CB is going for this blue-yellow, and CR is sort of going for this red-green, right? So that's kind of modeling the human vision. And that's why we are splitting it in these three independent channels, because that's what we think our eye sort of works with. And so again, if you have this image, this is how your Y, CB, and CR might look like. And we said that, oh, these still have a lot of correlation, right? So this explains why we did the color transformation from RGB to YCBCR. But if you recall, we also talked about downsampling, right? And then I said something that, oh, like you actually downsample the colored components more than the Luma ones because we care more about the average intensity than the everything else. So that's the other reason for actually even converting your RGB signal into these three independent channels, because these three independent channels are behaving differently. You care more about Y than CBCR. And that again comes from human, okay? So really the two reasons for doing this color transformation is, one is like the perceptual color space based on this idea of opponent theory, like that lilac chaser or that dress thing. And the second main idea is that you have different contrast sensitivity for your color channels versus Luma channels. If you haven't heard this before, let me just explain the idea very quickly. What your contrast sensitivity says, this is like similar to saying humans can hear 20 hertz to 20 kilohertz. Similarly, there are certain spatial frequencies and certain contrasts. So you can think of contrast just as the variance around that point, like how much the level shifts when you change 1 pixel versus when you change 1 byte versus 2 byte versus 4 bytes, you are changing different levels of intensity. And so your contrast really is like a function of your contrast plus spatial frequency. So what I have here in this image is there are bars with increasing and increasing spatial frequency, okay? And as you go down, I have higher differentials between these bars, binary bars. For me, I can make out like bars maybe in this region. This red line is supposed to be for a typical human, average human. Everything within it you should be able to distinguish as like some lines, versus around here you should sort of see it as a gray area. Now for different people it might be different. For example, for me, I actually think can't see any of this region, can't see this region. So my eyes are not perfect, right? So like you have this different contrast sensitivity. And this is important because like again, like you can, what this tells us like whether you can distinguish two signals or not occurring at different spatial frequencies. Why do you care about this? Can somebody think about why would I care about this for designing compressors? Anyone? Not that hard again.